Dr. Jake Forster, who is a medical physics registrar at the South Australian Medical Imaging, specializing in nuclear medicine. He has a background in radiobiology and microdosimetry, with interest in quantitative imaging and dosimetry of radionuclide therapy. Jake completed his PhD in 2018-2019 and actually received an award for the best medical physics PhD in Australia and New Zealand. So Jake, over to you, and looking forward to listening to your lecture.
Thank you, Eva. I'll just share my slides. So yes, thank you, Eva, for the introduction, and thank you for inviting me to speak. Oh, sorry, let me share. There we go. Yes, thank you for the introduction and for inviting me to speak at this workshop on radiobiology held by ACOMP. So the topic that I'm tasked with teaching you today is MIRD formalism, which is the method that's used to calculate dose in diagnostic imaging, nuclear medicine procedures, and also therapeutic procedures. It's called MIRD because it was originally invented in 1968 by the MIRD committee. So MIRD stands for Medical Internal Radiation Dosimetry. And throughout this talk, there's going to be a few questions that I've got, or exercises. So if you're able to participate, I recommend you grab a pencil and paper. Or if you're watching a recording, you can pause it and do them like that. Like Eva mentioned, if you have questions, post them in the comment section if you're on YouTube, or the chat if you're on Zoom. And also note down the slide number. So I've got the slide number at the bottom right-hand corner of my slides. So the contents for the talk is, I'm going to go through the MIRD formalism, which is actually pretty straightforward. And this will take us into half of MIRD, which is biokinetics. And for that, tracer kinetic modeling will come in handy. We'll also look at dose factors, and I'll introduce the quantity absorbed fraction. We'll also look at some pretty pictures of anatomical computational phantoms representing humans. I'll describe limitations of MIRD, which are important to know. And we'll also look at how MIRD can be applied in the cellular or at the cellular level. So in internal radiation dosimetry, the basic aim is to calculate absorbed dose to organs from the administration of an unsealed radioactive source. So if you think about how you might approach this problem, there's a few challenges that might jump into mind. So one would be that the radionuclide can spend time in many different organs. Another would be that the activity, or the disintegrations per second, in any organ will change over time. And maybe the third would be this appreciation that a disintegration in one organ can deposit energy not only in that organ, but also in other organs. And it could do this by emission of gammas, betas, electrons, and so on. So straight away, we'll jump into it. So for this formalism, we're going to introduce this concept called a target organ. So we'll call the target organ the organ that we want to calculate the absorbed dose for. We'll call that H. And then all the other organs and all organs that the radionuclide spends some time in, we'll call those source organs. And so our job is to consider the dose to the target organ, H, from disintegrations in a source organ, S. So what we'll do is we'll define a dose factor, and we write it like this, dF. So the dose factor for the target organ H from disintegrations in S is the mean dose to the target organ H per disintegration in source organ S. We'll then write the number of disintegrations in the source organ as NS, and simply we find the dose to the target organ H being equal to this product of the number of disintegrations in the source organ and the dose factor for a given target organ H from the source organ S. And then we simply sum over all of the source organs. So this is just calculating the dose to the target organ from one source organ and then from another source organ and adding them up. We should also note that the target organ, H, can also be a source organ for itself in general. And so this is the MIRD formalism. And I've just noticed we've had someone accidentally write on the slide.
Apologies, Dr. Jake, could
That's
you,
okay.
from the top, if you can just change disable the annotate.
Sure. Clear. Disable annotation from others. Yeah, okay, I've selected that. But I'm not sure how to remove the existing annotation though. Clear all drawings. Okay, all good. I think we're good.
Okay, sorry.
No problem. So straight away, we're going to do an example calculation because this is a workshop and I feel like you guys would benefit from doing some hands-on stuff. So I've made up a radiopharmaceutical. So just a toy radiopharmaceutical. This particular one only spends time in 3 organs, the liver, the brain, and the blood. And I've listed for you the biokinetic data, so the total number of disintegrations in each of these organs. So that's listed in the top table. And they've also given you the dose factor values. So these are those values that are absorbed dose in the target organ per disintegration in the source organ. So the way you see these dose factor values presented is typically in a table like this one. So here we've got the target organs listed as rows, and then the source organs listed as columns. And the units here again are milligray per decay. So for example, this value in the dose factor table corresponds to the dose factor for the target organ brain per disintegration in the source organ liver. So for example, it's 2 × 10⁻⁷ milligray absorbed dose to the brain per disintegration in the liver. So what I'd like you to do is to calculate the absorbed dose to the brain. So maybe just think about how you would do that. And if you're on YouTube or you're looking at a recording, pause it and have a go. So this is the answer down here. So we look at the 3 different source organs, including the brain itself. And again, it's just a simple multiplication between the number of disintegrations in the source organ by the dose factor for the target organ from that source organ. Let's look a little bit more into the biokinetic data. So this is the number of decays or the number of disintegrations in the source organ. So how do we get that value in practice? So for this, I'd like to introduce this quantity called the time activity curve, or the TAC. So the time activity curve for a source organ S, we'll call it just AS, and it's a function of time. And in general, this time activity curve has an uptake region where the activity is increasing in the organ, and it's got a retention region where the activity then decreases in the organ. So since activity is decays per second, or decays per unit time in general, if we then integrate over time the activity, what we get is the number of decays. And if we integrate the time activity curve from time 0, which we'll call the time of administration of the radiopharmaceutical, all the way to infinity, then we get this quantity called the cumulated activity. And this is denoted by the AS with a tilde on top of the A. So for example, in this plot on the right, we have activity in becquerels, so decays per second, and time is in seconds. So the cumulated activity in this case will be the number of decays. But in general, you might see these time activity curves presented with different units. So the activity could be in megabecquerels, for example, and the time could be in hours. So in that case, your cumulated activity would have units of megabecquerel hours. But what's important to note is that the cumulated activity is just equal to the number of disintegrations in the source organ up to some choice of units. So this is really a dimensionless quantity, even though you can see it given with units. The next quantity I want to introduce to you has some negative connotations to it, but it is still used today. And the reason it has some negative connotations is because it's a common source of confusion for people in this area. So this is the residence time. So I want you to consider that green rectangle on the figure, which has height equal to A0, which is the administered activity. So the residence time for the source organ is simply the width of this rectangle, such that the area of the rectangle is equal to the area under the time activity curve, or the accumulated activity. That's all this is. Typically, this has units of hours. It'll be quoted in units of hours. And like I said, because people don't like using the term residence time, it's now sometimes called the time integrated activity coefficient. But all it is really is this constant of proportionality between the accumulated activity and the administered activity. So the residence time is defined as the accumulated activity in the source organ divided by the administered activity. So it's important to note that that is the administered activity A0 and not the initial activity in the source organ. If you go and look at some publications in internal radiation dosimetry, you can see this MIRD formalism written a couple of different ways. So the way that I've presented it to you so far is actually called the radar way of writing things. And radar stands for Radiation Dose Assessment Resource, so that's their own group. And so this is the formalism in which we talk about the number of disintegrations and multiplying that by a dose factor, which is in units of milligray per disintegration. The original way that the MIRD formalism was presented is actually using the accumulated activity and this S factor. So instead of writing the dose factor, they called it the S factor. And the only difference really is that the accumulated activity usually has units, say megabecquerel hours. And then so the S factors are presented in units of milligray per megabecquerel hours, say. So this is a little bit more practical if you're using MIRD practically, but it doesn't really convey the underlying meaning as well as the radar way. And in the MIRD formalism, they can also write the MIRD formalism in terms of the residence time in the source organ and the administered activity, and that's shown there as well. But importantly, these are just the same thing. Now, since this talk has diagnostic imaging in its name, I feel like I can't pass up this opportunity to talk about how we determine risk of detriment from stochastic effects in diagnostic imaging. So the MIRD formalism will give us organ absorbed doses, and we'd like to go and predict the risk of detriment from stochastic effects from those organ absorbed doses. That's the aim. And by stochastic effects here, we're talking about cancer, so the induction of cancer, and we're also talking about genetic effects that may occur from gonadal exposures. So again, if you think about how we might go about this problem, there's a few things you'd consider, but certainly one of them would be the type of radiation that deposited the energy. And you might also consider which organs or tissues were irradiated. So as you've heard from previous speakers during this workshop, the absorbed dose isn't the only thing that matters if you're looking at radiobiological effectiveness. It also matters how the absorbed dose was deposited. And so I'm thinking here along the lines of LET. So for this, we have this quantity called the equivalent dose. And the equivalent dose is defined for a particular organ. The equivalent dose to an organ is defined as the product of the absorbed dose to the organ from a particular radiation type R multiplied by the radiation weighting factor for that radiation type, and then summing over all of the radiation types that deposited dose. So this equivalent dose has units of sieverts instead of gray. And so these radiation weighting factors have units of sieverts per gray. And these radiation weighting factors, you can generally think of them as increasing with increasing LET of the radiation type. So for example, for photons and electrons, we take them to be 1. For protons, we take it to be 2. For alphas, 20. And for neutrons, we use a continuous function of energy. So I've got a quick example calculation there. If you had a liver receiving 2 gray from X-rays and 3 gray from protons, then the equivalent dose to the liver would be 8 sieverts. So the next thing we want to take into account is that different organs, if they are irradiated with the same equivalent dose, then it might not confer the same risk of detriment from stochastic effects, depending on which organ received that equivalent dose. So for this, we have this quantity called effective dose. And the effective dose is only defined for the whole body. And we attain the effective dose by multiplying our equivalent dose to some organ or tissue T by the tissue weighting factor for that tissue, and then summing over all of the tissues that received an equivalent dose. Unfortunately, the effective dose also has units of sieverts, so it can be a little bit confusing if you see a quantity in sieverts out of context. We have this property that the tissue weighting factor, they sum to 1 for all of the tissues in the body. But importantly, the effective dose to the whole body by design, I should mention the tissue weighting factors, they're larger for tissues that are either more radiosensitive, so perhaps cancer is more likely to be induced in that tissue from an exposure or from a given equivalent dose. But they're also higher if the prognosis with a cancer from that tissue is worse, or if the quality of life living with a cancer from that tissue is worse. So by construction, the effective dose is actually proportional to a population-based risk of detriment from stochastic effects.
All users can now please log off for 60
Sorry, I'll just
seconds
pause for a second.
and log on again to commence use of Sunrise EMR. Thank you.
Thank you. So yeah, the effective dose is proportional to a population-based risk of detriment from stochastic effects. Having said that, we can still use the effective dose to predict a risk of fatal cancer. So we go roughly, it's about 5% risk of fatal cancer per sievert of effective dose. And this kind of risk coefficient, or this risk model, actually comes from data from the atomic bomb survivors. And that data is actually only above about 100 millisieverts. And the extension of this risk model of 5% per sieverts to below 100 millisieverts is called the linear no threshold model. But it's important to stress that this risk coefficient of about 5% per sievert of effective dose is a population-based risk. It's not actually the risk to any individual, any particular individual. So back to MIRD. What is there to do now that we have our formalism? So we really have two separate jobs. One of them is to obtain a description of the time activity curve that we can then integrate over time to obtain our cumulated activity, our total number of disintegrations. And the second job we have to do is obtain these dose factors. So this mean absorbed dose to a target organ H per disintegration in a source organ S. So that's our two separate jobs. So we'll start with the biokinetics, the first one. And because I'm not sure about background of some of the viewers, I'm going to talk about effective half-life just quickly. So you might have got this, you might have this perception that if you have activity in a source organ, it's removed not only from physical decay, but also from biological processes. And what we do is we assume that the biological clearance is also exponential, and it has a half-life equal to, or we denote the half-life by T for B for biology or biological, and S for this, whatever the source organ is. And we can give it a decay constant as well, just from the exponential. And then we have to appreciate that the activity in the source organ will reduce due to both biological clearance and physical decay. And because of that, we have this effective decay constant. So in this event of two exponential processes happening at the same time, it's the decay constants which add. So we have an effective decay constant, which is the sum of the physical decay constant and the biological decay constant. And we can also define this effective half-life the normal way for an exponential. And we also note that in this case where biological clearance is very slow, or you have a long half-life for biological clearance, then the effective half-life is just essentially the physical half-life, which makes sense. I've got another example problem. We've got a female patient who receives an intravenous administration of 500 megabecquerels of a technetium-99M labeled radiopharmaceutical. 30% is taken up by the liver and 70% by the kidneys. The uptake is rapid and there's negligible uptake in any other organ. We have the clearance from the tissues taking place with a biological half-life of 12 hours. And there's no biological elimination from the liver. So we've got two jobs. One would be to calculate the effective half-life in each of the above organs. The second would be to then calculate the accumulated activity in each of the above organs. So the kidneys and the liver. Pause it if you're watching a recording and have a go. So the first thing to note is because uptake is rapid, we'll have no uptake region in either of the time activity curves for the organs. So the time activity curve will simply be exponential with some effective half-life. We'll use that equation presented in the previous slide to calculate our effective half-life. We can simply write the time activity curve in this kind of fashion, where the initial activity in the organ, we can get that from those uptake values. And then all we have to do to get the accumulated activity is to integrate over an exponential, which is quite straightforward, and we get a nice answer. So it's not always easy to measure a time activity curve for some tissues or some organs. So some tissues or some parts of any system, especially biological systems, aren't easily accessible by measurement. So for example, you could attain activity measurements or activity concentration measurements for blood by taking blood samples. For certain organs, you could obtain activity measurements for the organ by doing quantitative imaging. But it's not clear to me, at least, how you would, in an image, distinguish between activity that's in the wall of an organ versus in the contents of an organ. So example organs for this would be like bladder, heart, stomach, intestine, gallbladder. Similarly, how would you distinguish between activity that's in the bone matrix or in the bone surfaces on an image? So this is really the motivation for these tracer kinetic models, is that we want to make measurements in the accessible portion of our system, and then we use our tracer kinetic model to estimate parameters for inaccessible features of our system. So in compartment models, obviously we have this term called compartments. So what are compartments? They're volumes or spaces in which a tracer concentration will rapidly become uniform. And so for us, we're going to be considering compartments to be organs or tissues. Let's denote the concentration of a tracer in a compartment A with CA as a function of time, T. We're going to define the flux or the flow to be the tracer concentration that's moving from one compartment to another per unit time. And generally, we'll just consider what's called first order kinetics. And in first order kinetics, we have the flux that leaves a compartment being proportional to the instantaneous concentration in that compartment. So for example, the flux leaving compartment A will just be proportional to the concentration in compartment A at that time. Or if you like, the flow from compartment A to compartment B is only proportional to the concentration in compartment A. So I've got an example of a model on the right. This is just a two-compartment model, compartment A and compartment B. And we can write down this equation for the time rate of change of the concentration in compartment A. And it's just going to be equal to the flow from B to A minus the flow from A to B. So what's flowing into A minus what's flowing out of A will be the concentration in A per unit time. So like I said, the flow from B to A will only be proportional to the concentration in compartment B. So that's what we've written down there. And then the flow from A to B will only be proportional to the concentration in compartment A. And so this equation here, or that picture even, is what we call a tracer kinetic model. So these K1 and K2, they're called rate constants. They're simply in this case, the fraction of the compartment's concentration that's leaving the compartment per unit time. And if instead of considering concentration, we were to write this equation in terms of activity, all we would need to do is add one extra term, which to account for physical decay. And of course, that's going to just be proportional to the activity in that particular compartment. So our next exercise, I've got another compartment model. This is a three-compartment model. I'd like you to write down the time rate of change in the concentration of compartment B. Again, pause it if you're watching a recording. It might look hard, but it's actually not very hard. So all we have to do is consider the flow from A to B. That's going to be going into B. And then we have to subtract off the stuff that's flowing out of B. So we subtract off the flow from B to A, and we subtract off the flow from B to C. So that's what it looks like. That's the equation. Now, these tracer kinetic models can get very complicated if you're looking at real biological systems. They can get very large. So for example, this is the compartment model for iodine. However, in general, the tracer kinetic model, it's actually usually very easy to write down if you're considering first order kinetics. So for example, the general equation is shown there. So the time rate of change, the activity in the compartment I can be written like that. And the very first term is the activity flowing out of compartment I. The second term is physical decay. And then the summation and all of the other terms are activity that is flowing into compartment I from compartment J. So even though these equations can look very complicated, the good news is that under certain constraints, the solution's actually pretty straightforward. In particular, if we assume immediate uptake in the source organ S, then we can write the time activity curve, or in this case, the time activity curve divided by the administered activity, as a sum of exponentials. So here we've got, again, A naught is the administered activity. Fs is the fractional distribution to the source organ S. And then for each of these exponentials, we have the parameter AI, which is like the fraction of Fs that gets eliminated with an effective rate constant, lambda EI. And interestingly, these AI parameters can be negative because essentially what you're doing at this point is curve fitting to your data. But importantly, they all sum to 1. So now that we have a time activity curve, we have an expression for a time activity curve, we can just integrate this from time 0 to infinity to get our cumulated activity, or in this case, we'll get our residence time because we divided by the administered activity. And we have a very straightforward equation for our residence time in the source organ S. ICRP 128 is a very useful document for looking up doses from nuclear medicine diagnostic imaging procedures. And the cool thing about ICRP 128 is they actually give you the information about the biokinetic models that they're using in their dose calculations. So the biokinetic information is presented in tables like the one shown here. So this is the table for FDG, for example. And the column on the first column on the left is the source organ, S. The columns 2 through to 4 are these parameters that I showed you in the last slide. So these parameters are the ones that describe the time activity curve in the source organ S, and they're the ones that are used to calculate the residence time, like I showed. And that last column is the residence time in the source organ S. But what most people in practice do when they use ICRP 128 is they just look at these tables. So these tables give you absorbed dose in a particular organ per unit of administered activity. They also give you the effective dose per unit of administered activity. So that's one half of MIRD. The other half is these dose factors. So recall the dose factor is the mean dose to a target organ H per disintegration in source organ S. So how would we actually measure a dose factor? So the first step is to consider each emission, we'll call them I, from the radionuclide separately. So by emissions, I'm talking about, for example, the gamma that results from a transition between 2 daughter energy levels, or the beta for a decay to a particular daughter energy level. Those would be separate emissions. And for each of these emissions, we're going to define an absorbed fraction, phi, again, as the fraction of energy emitted from the ith emission in source organ S that is absorbed in target organ H. So then if we just take the product of our fraction of energy emitted in the source organ S that is absorbed in target organ H, and we multiply that by the energy that was emitted per disintegration due to the ith emission, then we get the total energy that gets absorbed in our source organ H per disintegration. So then we just need to divide by the mass of the organ H, the target organ H, and we get the dose to the organ H per disintegration in source organ S, after we sum over all of those emissions. So this equation can look a bit complicated, but if you stare at it long enough, it should make sense. Oh, so yeah, the energy emitted per disintegration due to the ith emission is simply the product of the yield, so the number of emissions per disintegration, and the average energy of the ith emission. So in practice, these absorbed fractions for a given source organ S are actually typically presented in tables with target organs as rows, like shown below. And instead of doing it for each emission, they actually just, at least in the old days, they gave it in terms of discrete photon energies and let you sort of interpolate between them to get your gamma emissions. We also have this general kind of approximation that for photons that are very low energy, like less than 10 keV, also for electrons, for betas, and for alphas, we can assume that the source organ is the organ where all of the energy is deposited. So this absorbed fraction is 1 if the target organ is equal to the source organ, and it's 0 if for other organs. And the absorbed fraction has this cool property where you can calculate the inverse, or the inverse is equal to itself. So the absorbed fraction for some emission for a given source organ and target organ combination is the same if you switch the target organ and the source organ. This gives us a nice relationship between the inverses for the dose factor as well, just being related by the ratio of the masses. And you'll also see a quantity called the specific absorbed fraction, which is just the absorbed fraction divided by the target organ mass. And you can also see the dose factor equation written in terms of the specific absorbed fraction like that. So in order to actually measure an absorbed fraction and hence a dose factor, we need to actually do Monte Carlo simulations. So one of the things these simulations has to have is a phantom that represents the anatomy of a human being. And basically the procedure for measuring an absorbed fraction or a specific absorbed fraction would be like shown here. So firstly, we assume all the disintegrations are uniformly distributed in a source organ. And then we would just simulate n emissions of type I in that source organ. And then we'd score the total energy that gets deposited in some target organ H. And then the specific absorbed fraction for the target organ H from the emissions in the source organ S will be given by this ratio. So it's that total energy deposited. We'll divide by those number of emissions that we did. So that's like total energy deposited per emission. If we multiply by the yield for the ith emission, that'll give us the total energy absorbed per decay. Then we need to divide by the energy of the emission, EI, and that'll give us the absorbed fraction. And then we'll divide by the mass of the target organ, and that'll give us the specific absorbed fraction. So there are two components to these Monte Carlo simulations that I'm going to talk about. The first will be the track structure. So Monte Carlo track structure, there's a few considerations that you can consider. The first would be whether you're going to use condensed history processes to simulate your tracks, or whether you do step-by-step tracking. So condensed history processes, that's where you look at a cumulative effect of multiple electron collisions in a single step. Whereas step-by-step or event-by-event tracking is where you consider each event explicitly. So for example, that figure on the right, Livermore physics is an example of condensed history processes, and GM4 DNA physics is an example of step-by-step tracking. Important to note here, though, is the scale of the figures. So this is across 15 nanometers. So step-by-step tracking is really only appropriate in the microdosimetry domain, or at the most inside of a cell, the cellular level. For us, for our purposes, for the Monte Carlo track structure simulations for macrodosimetry, like in MIRD, we probably don't need that. Condensed history is probably okay. Another thing in Monte Carlo track structure to consider is these interaction cross sections. So we're talking about probability for events like electron ionizations, excitations, bremsstrahlung, all the gamma processes. The other thing is the cutoff energy. So how far down do you track these electrons? Say at what energy do you stop tracking, and do you just dump all the energy of the electron where it stands? There are lots of examples of Monte Carlo track structure code. I've listed some of them there, and some of them specialize in certain energy domains, whereas others are quite general purpose. But I think the cooler thing about these Monte Carlo simulations is actually the phantoms, so the geometry side of things. The first ever dose factors were calculated for these stylized phantoms by Schneider et al. In about 1975. And these phantoms use what's called geometric primitives, which basically just means all the shapes you're familiar with, like cones, spheres, ellipsoids, cylinders, things like that. And they, to get their organ sizes and shapes and so on, they used information from ICRP 23, which is a report on the reference man. So they gave organ sizes for the average adult worker of the Western Hemisphere. So yeah, Schneider simulated that, and that was the first ever phantom for which dose factors were calculated. It's also called the MIRD phantom. Much later, more than 10 years later, we had Christian Eckermann extend the MIRD phantom to a series of pediatric phantoms as well. So he did phantoms for a newborn, a 1-year-old, a 5-year-old, a 10-year-old, and a 15-year-old. And these phantoms are actually hermaphrodites, so they have both male and female sex organs for the purposes of calculating dose factors. And interestingly, it was practice back then was to use the 15-year-old as a surrogate for the adult female. And I think most notably is the values in ICRP 128, those dose values that we saw in the table, they're actually calculated using these phantoms. So it wasn't until 1995 that we finally got a dedicated purpose-built female adult phantom, and that was by Stabin et al. So that finally came in 1995. And we also got 3 pregnant phantoms. So 1 3-month pregnant, a 6-month pregnant, and a 9-month pregnant phantom. These were very important for doing dosimetry for calculating fetal doses from in utero exposures. In the last 20 years, though, we've had a different kind of phantom. So all of those phantoms were based on the geometric primitives. But in the last 20 years, we've had a new generation of phantoms called boundary representation phantoms. There's really 2 types. One is non-uniform rational basis spline, or NURBS phantoms, and the other is polygonal meshes. But collectively, we'll just call them boundary representation phantoms. In 2001, Segars produced the first ever boundary representation phantom, and this was NURBS-based. This is a cardiac torso phantom, or the NCAT phantom. And one of the coolest things about the boundary representation phantoms is the shapes in them are very easy to deform. So in particular, he made a 4D phantom. So he made a phantom that can produce cardiac and respiratory motions. This does have limitations though, so it's a torso only, it's a male only. And because it was purposed for nuclear medicine, the structure is actually quite low resolution. About 10 years later, he reduced an extension on his NCAT phantom, the XCAT phantom. And this phantom is much more higher resolution. So for example, the brain alone has more than 100 vessels and structures. And it's also a whole body phantom. He also released phantoms for lots of different BMIs, ages, and so on. So it's not just a single phantom. But the first dose factors produced for a boundary representation phantom was produced by Stabin et al. In 2012. He used the NURBS phantom from Segars, but he used the size data from a new ICRP report, ICRP 89. And he's produced dose factors for adult male, adult female, pediatrics, and pregnant women. These phantoms are pretty impressive. So now let's talk about some limitations of MIRD. So I've put up the equation for the MIRD formalism again for your reference. And let's look at the 2 separate components, the biokinetics components and the dose factor components. We'll look at those separately. So the dose factor, so that absorbed dose in target organ H per disintegration in source organ S. Let's look at some of the quantities that are in that. So first we have those average energies and yields of the emissions. That comes from nuclear decay data, and the uncertainty in that is pretty small relatively. So that's okay. That's not a great source of uncertainty for us. The absorbed fractions, or the specific absorbed fractions, and the organ masses, it's important to note that these values are always for a reference phantom. So regardless of how realistic the phantom is, even if it's a really cool boundary representation phantom, that phantom will not match most patients in general. You also saw when we walked through the Monte Carlo simulation that we have to assume the disintegrations are spatially uniform in the source organ, which again isn't what happens in reality. So there's a couple of limitations. On the biokinetic side of things, we can break our total number of disintegrations in a source organ. We can break that down into the administered activity and the residence time. So the administered activity, we measure that in a dose calibrator. And if you've got a good QC program for your dose calibrator or QA program for your dose calibrator, the uncertainty in that is usually not too bad. But the residence time, on the other hand, if you're using, for example, ICRP 128's tables, dose tables, all of those residence times come from compartment models. And the model parameters are often estimated from animal studies, not even from human studies. But even if they were from human studies, we still have to appreciate that there's a large variation in biokinetics between individual patients. So that's a very large error, or a very large uncertainty in that particular quantity. So we have this great quote from ICRP 128 itself that says, estimates of absorbed dose to different organs will not generally deviate from actual absorbed doses in patients by more than a factor of 3. And the effective dose may vary within a factor of 2. So that's the kind of uncertainties that we're dealing with in nuclear medicine. I've got a couple of plots here that are showing time activity curves for 2 different organs, urinary bladder and gallbladder. This comes from a paper from 2009, but you can just see, especially for gallbladder, how much between different patients, how much variation there is in the time activity curve and ultimately the residence time in that organ. This is the major source of error for us. And just finally, I'll show you briefly MIRD in the microdosimetry context. So for this whole talk, we've been talking about tissues and organs as being the sources and the targets. We can just equally apply this formalism in the cellular domain. So we can consider cells to be, 1 minute, Eva, last slide. We can consider cells to be the target or the source, the whole cell, or we can even consider components of the cell to be source or target regions. So typically in this microdosimetry, we would do a multicellular geometry, and the biokinetic data is similar, except instead of cumulated activity in an organ, we're considering it in the cell or in a cellular component. This is typically done by sampling from a distribution. And for the residence time, instead of defining it in terms of the administered activity, we would do the initial activity in the cell. And interestingly, the dose factors, in the cellular context, they separate the dose factor into a self-dose and to a cross-dose. So self-dose is the dose factor for disintegrations in the same cell, whereas cross-dose would be a dose factor for disintegrations in a different cell. The reason this is done is because the dose from disintegrations in the same cell versus in other cells can be deposited with different LET values. So if you're importing your dosimetry into a radiobiological model, you might want to have those 2 components separated and treat them separately. So some examples are MIRD cell and paradigm. MIRD cell is shown there. So just to conclude, the MIRD formalism is very powerful, it's very elegant and a general method for calculating dose from radionuclides in biological systems. The biokinetics and the physics, or the dose factors, are treated separately and they're completely independent. I think that's why it's elegant. We have these tracer kinetic models or compartment models, which we can use to derive biokinetic data, which we can't access by measurements. And we do Monte Carlo simulations with anatomical phantoms to obtain the dose factors. So this is the method used to estimate dose from diagnostic nuclear medicine procedures. And we like to use those tables from ICRP 121 or 128. But we have to appreciate that dose calculated in this way, dose received by an individual, may differ considerably from the ICRP 128 tables, owing to patient divergence from the reference phantom geometry and also the reference biokinetic data. Thank you very much.
Your yes, your test.
My
Paul,
test?
Jake, yes, next
Oh.
slide.
Do we have that to put up in Zoom?
Yes,
Here we go.
but also put it on your,
Okay.
do next slide, please.
Yeah.
Yeah, so that people on YouTube can see as well.
Okay.
Okay.
Can everybody see the poll up onto their screen?
Yes. Okay, we can stop polling question 1. Okay, so the correct time was residence time. Jake,
Yeah.
can you explain one?
Yeah, so the majority got it right. So the residence time is the cumulated activity, or sorry, the residence time is the total number of disintegrations divided by the administered activity. Whereas the area under the time activity curve and the cumulated activity, they're both equal to the total number of disintegrations.
Yeah, yeah.
If you have the correct units.
Next question. I thought we have one more. Is that the only one that we've got there? Okay, here we are. Okay.
So I think it didn't come through very well.
Yes, can you read it for us?
The second one should be 0 to the energy of the emission.
Mhm.
I like that some people put that as their answer there.
Well, I think we can see the slide. So on the slide, we can see the right wording behind.
Oh, true. Yeah.
Yeah. Okay, we can end polling.
Good. The majority got it, just.
Yes.
So the absorbed fraction can be as little as 0, but it can't be any more than 1.
Because it's a fraction of 100%. Yes.
Yeah, exactly. Yep.
Yeah. Wonderful. Thank you very much, Jake.